Sorry. All right, so today we're going to get started with looking at regulation of gene expression. So now we know how we go from a gene in our DNA to create a protein through protein synthesis. But that is, at most only half of the story because after the Human Genome Project took place in the late 90s, scientists realized that there were far fewer genes than they thought on all of the human chromosomes. And one of the reasons that, you know, humans being so complex organisms could still be working in the way that we know that they do, have the physiologies that we know that they do, and able to respond to a variety of changes in external and internal environments, but only have as few genes, about 20,000, that they do, is because we are able to regulate or control how those genes are expressed, when and to what extent and under what conditions. Um, and that's why now, in present times, studying gene expression and the ways it's regulated and controlled is very important for our continuing understanding or the evolution of our understanding of genetics. So we will get to eukaryotic gene expression, so like us, tomorrow. But to get some of the basic ideas, I wanted to talk about control of prokaryotic, so bacterial gene expression first, just because it's a little bit simpler, but a lot of the fundamental ideas that are key in eukaryotic gene expression are shared in prokaryotes as well. So just like any other organism, bacteria need to respond quickly to changes in their environments, whether that's external environments or internal environments. In prokaryotes, since there is no mRNA processing, post-transcriptional processing, transcription and translation happen at the same time or simultaneously. Um, so when prokaryotes regulate the expression of genes, that's mainly transcription that they're regulating. So one example that we're going to be talking about is the regulation of metabolism. Um, and that's because it's been a very deeply studied, easily illustrated example of gene regulation in prokaryotes. So if we look at this picture on the right, it is an example of maybe how a prokaryote synthesizes tryptophan, which is a necessary amino acid, from precursor materials. And you can see that there's several steps, each catalyzed by a different enzyme that is resultant from a specific gene, from the precursor material to tryptophan. And in prokaryotes, instead of blocking the enzyme function, uh, one at a time, um, instead there's a block in the transcription of genes for all the enzymes in the tryptophan pathway. This is important because it saves energy by, not wasting uh, energy and resources on making unnecessary protein synthesis that isn't needed. So as we look here, tryptophan is an inhibitor, so it prevents enzyme number one from working, and it also prevents the transcription of all 5 genes that create the enzymes 1 through 5 that break down, or not break down, but lead to tryptophan biosynthesis. Cells in prokaryotes vary amount of specific enzyme by regulating gene transcription. So turning it on and off, or speeding it up or slowing it down. Um, so as an example of why a gene might be turned off, if a bacterium has enough tryptophan, just from its natural environment, then it doesn't need to make enzymes that are used to build tryptophan. That would just be a waste. So if there's plenty in its external environment, we can turn off the genes that are responsible for creating enzymes that would build tryptophan. Another example of why a gene might need to be turned on is pretend a bacterium encounters like a new sugar, which you can think of as a potential energy source, maybe like lactose. Then it needs to start making enzymes that would be used to digest that lactose. It wouldn't make any sense to produce those enzymes if it was an environment without lactose. But once it is, it would want to turn those genes on so that those enzymes used to digest lactose are being transcribed and translated. The main way in which this on-off switch is accomplished in prokaryotes is through this idea of an operon, which is that genes are grouped together that have related functions. So, for example, all the enzymes in that tryptophan pathway would be grouped together, all of the genes for all those enzymes to make tryptophan, or all of the genes that went to make proteins or enzymes that would digest lactose would be grouped together. And that grouping is called an operon. Okay? There are two types of operons called inducible and repressible operons. We'll talk about in just a little bit. But first, we want to take a look at general operon structure. So each operon has a promoter, and this is the same thing that we talked about when we looked at transcription. It's a binding site for RNA polymerase. So that's kind of like the TATA box we've talked about. A single promoter controls the transcription of all of the genes in an operon. So it's either completely turned on, all the genes, are completely turned off. So they're transcribed as one unit, a single mRNA for all of those genes in a prokaryote. And then there is slightly downstream, so slightly closer to the DNA that would make up all of those genes in the operon, something called an operator, which is a DNA binding site for a repressor protein, a special protein that might repress or stop the transcription of that gene. Okay, and on the next slide, we're going to see a good picture to start to get an intuitive understanding of what this means. So here's the operon model. It contains those essential parts, the operator, the promoter, and the genes they control. And it really is the model for gene regulation for prokaryotes. So let's take a look. So here's the promoter in green. That's where RNA polymerase is going to be attracted to from all the transcription factors. That's what the TATA box is, the start point. Okay, and then slightly downstream, we call this way downstream. Maybe I'll write that on the screen because I use these terms a lot, and in the literature, you'll hear the terms a lot. So this is what is called downstream, this direction closer to the genes, and this is upstream, farther away from the genes. So downstream from the promoter is the section called the operator, where down at the bottom of the screen, this repressor protein will bind. And the repressor protein is what turns off the gene by binding to that operator, and it blocks the RNA polymerase. It doesn't let it go through. It prevents it from binding and moving down the DNA to transcribe those genes. So without the repressor protein active, the RNA polymerase can move down the DNA and transcribe those genes into mRNA, and then those mRNA will be translated into proteins for those 4 enzymes. So when it is turned on, this is what will happen. When the gene's turned off, the repressor protein will bind to the operator, and the RNA polymerase can't bind to the promoter, and this transcription won't occur. So we don't make any mRNA, and we don't translate that into any proteins. So that's the general model. Let's talk about 2 variations on that, which are called the repressible and the inducible operons. And these are things that you will need to be very familiar with. So here's an example of a repressible operon. Like let's just call this the tryptophan operon. And it's a synthesis pathway. Um, when excess tryptophan is present in the environment, the tryptophan binds to the tryptophan repressor protein. So here's the repressor protein. Here's the tryptophan. And when the tryptophan binds to the repressor protein, it activates the tryptophan repressor to make a tryptophan repressor protein complex. Okay, so a repressible operon, the repressor is inactive normally. Let's say at the null state, at the normal state, the repressor is inactive, which means that normally RNA polymerase is able to bind to the promoter, move past the operon, and transcribe all of the genes. But when tryptophan is present, that's the stimulus, tryptophan will activate the repressor protein and cause it to be able to bind to the operator, blocking RNA polymerase binding. So normally, when tryptophan isn't present, RNA polymerase is able to transcribe the genes and they can be translated into enzymes, right? That's what normally happens. So normally, as the normal null state, it is on. A repressible operon is turned on. The repressor is inactive. When we have the stimulus of tryptophan being present, like this, now we have a bunch of tryptophan, it activates the repressor protein, which binds to the operator and prevents RNA polymerase from binding. So in a repressible operon, the genes are normally turned on, but they can be turned off if a specific condition is met. Tryptophan is prevalent in the environment. So repressible proteins, the genes are normally turned on, the repressor is normally inactive, unless a specific condition is met in which the repressor is activated and the genes can be switched off. So here is another way of looking at the same idea. This is a picture that's used in your book, and it's just saying kind of the same thing. Okay, the repressor protein is always present, and tryptophan, which we call the co-repressor, it's what activates, turns on the repressor in a repressible operon. Uh, when present in high quantities, is able to activate the repressor and prevent RNA polymerase from binding. Now, on the other hand, we have what's called an inducible operon. This might happen like in our example with lactose, the digestive pathway. When lactose is present, lactose binds to the lac operon repressor protein and triggers its release from the DNA. So, in this case, the null states, normally this gene is switched off. Normally, the repressor is always active and always bound to the operator. And so normally RNA polymerase can't bind to the promoter and the genes can't be transcribed, kind of in the everyday state. Now, when a certain condition is met, a certain stimulus, which is lactose, is prevalent, lactose will bind to the repressor and deactivate it to shut it off. And the repressor will fall off of the DNA, and RNA polymerase will be able to bind the promoter and transcribe those genes into mRNA, and they can be translated into proteins. All right. So an inducible operon is normally off, but can be induced, can be caused to turn on if the certain stimulus is met, which in this case is lactose is present in the environment. So here's another example. In inducible operons, we call the substance that binds to the repressor, this time to inactivate it, the inducer. That's why it's called an inducible operon. So in these examples with lactose, we can upregulate or increase the rate of transcription by turning it on and off. Right? We can use secondary messengers to do this, to increase the rate at which transcription is occurring. We can use enhancers like we talked about with transcription a couple days ago. But in general, we want to know the idea, the basics for today is to understand the difference between repressible operons and inducible operons, examples of their situations where they'd be turned off and on, and then the general structure of an operon. Okay, so that's going to be it for today, and we'll go over this here in class tomorrow.